Hello and welcome to the Cell Culture Laboratory. In this video, I will show you step by step how to passage the L929 cell line. First, you will see how to prepare the workspace and materials under the sterile workbench. Next, the video demonstrates how to remove the old medium and wash the cells with PBS. After that, trypsinization and morphological inspection under the microscope are performed before neutralizing the trypsin and centrifuging the cells. In the following section, you will learn how to remove the medium containing trypsin, add fresh medium, and fill the culture flasks to a total of 5 milliliters. Finally, the video covers cell storage after passaging, a recheck of cell morphology, and the cleaning procedures under the sterile workbench. Let's look at each step in detail.
The passaging. Prepare materials and workspace. Remove medium and PBS from the refrigerator and pre-warm in a water bath. Before starting, all necessary tools should be laid out and disinfected to ensure a sterile environment. Choose the appropriate pipette size for precise and efficient work. Pipettes and other tools should be lightly wetted with disinfectant before use. Only a thin, even coating of disinfectant is needed. Too much can form droplets or puddles that contaminate sterile areas. Cell culture flasks have a filter in the lid for gas exchange. During transport, ensure no medium enters the filter to avoid contamination. Trypsin is stored frozen and must be completely thawed before use. Otherwise, the concentration is incorrect and dilution will be inaccurate. Regularly clean the sterile workbench glass and work surface with disinfectant to prevent contamination. Gloves must cover the lab coat sleeves or arm cuffs should be used. Exposed skin or wrists increase the risk of contaminating the sterile zone. Open all flasks and place the lids on top to allow one-handed opening later. Removing old medium. When working with culture flasks, do not touch the cell layer with the pipette to avoid detachment or damage. Always hold the lid in your hand. Never set it down. Aspirate the old medium with a pipette and transfer it to a waste bottle. Dispose of the used pipette properly. Only the cells attached to the flask wall remain. Washing cells with PBS. Wash the cells with approximately 3 milliliters of PBS to remove residual medium. Gently swirl to ensure all cells are covered and clean. Remove the PBS. Never leave cells without medium for too long to avoid drying out and cell death. Before use, pipette trypsin up and down several times to ensure a homogeneous solution after thawing. Add 1 to 1.5 milliliters of trypsin to detach the cells from the surface. Incubate briefly, at least 5 minutes at 37 degrees Celsius, to allow trypsin to break cell connections. Never touch the lid or filter with your fingers, as this can introduce contamination. Handle the lid only at the edges and within the sterile zone. Incubate the cells for 5 to 10 minutes in a CO2 incubator for effective trypsin activity. Morphological control. Remove the flask from the incubator and check the cell's appearance under the microscope. If cells remain attached while gently moving the flask, trypsin has not fully detached them. Tap the flask gently to release remaining attached cells. The bottom of the flask should show a milky suspension containing detached cells. Disinfect and clean the workspace after use. Neutralizing trypsin. To neutralize 1.5 milliliters of trypsin, add approximately 3 milliliters of fresh medium. Gently rinse the growth surface with fresh medium while adding. Centrifugation. Transfer medium with cells into a 15 milliliter centrifuge tube. Close the lid and label the tube. Prepare a counterweight tube with 6 milliliters for balance. Centrifuge cells at 330 times G for 7 minutes. The cells will form a pellet at the bottom. After centrifugation, the pellet contains the cells separated by centrifugal force. Removing trypsin-containing medium, carefully aspirate the supernatant without disturbing the cell pellet to avoid cell loss. Dispose of used materials properly. Add fresh medium. For continued culture, add 4 milliliters of fresh medium to 1 milliliter of cell solution, giving a total of 5 milliliters in the centrifuge tube. Resuspend cells gently by pipetting up and down to evenly distribute cells, break up clumps, and create a uniform suspension. Seeding flasks. Distribute the cell suspension evenly across 3 fresh culture flasks, 1 milliliter per flask. Add medium before adding cells to ensure even distribution and optimal adherence. Adding cells first increases the risk of uneven growth and contamination. Then add 4 milliliters of fresh medium per flask to ensure optimal growth conditions. Hands should not cross the front ventilation grills of the sterile bench, as unfiltered air could disturb the sterile zone. All work must be performed within the airflow area. Never immerse the pipette tip into the flask. Add liquids carefully along the edge without touching inner surfaces. Close and correctly label the flasks. Remove any droplets, especially cell suspension, immediately. Moist areas on the sterile surface are a breeding ground for microorganisms and can compromise sterility. Storage after passaging. Place culture flasks in the CO2 incubator to allow cells to attach to the bottom. Morphological check. After approximately 24 hours, cells are usually well attached and spreading. To form a confluent monolayer, cells typically need 2 to 3 days of incubation. Final procedures in the sterile bench. After completing work, clean and disinfect the workspace thoroughly. Close the sterile bench and turn on the UV light for disinfection of remaining items and surfaces. Leave the UV light on for at least 1 hour to ensure microorganisms are effectively eliminated.